Hey everybody, it's Mr. Poser, your human anatomy and physiology teacher. This is going to be our third video in our first unit in human anatomy and physiology on our biology review. And we're going to go over the basics of the cell membrane today because the cell membrane is going to be very, very important, or an understanding of the cell membrane is going to be very, very important when it comes to our central understandings of a lot of the organ systems and a lot of the tissues that we're going to see in human anatomy and physiology. All right, so let's get started without further ado. Overall, the function of the cell membrane, it controls what goes in and what goes out of the cell. That's a very basic definition. It has a lot of other functions, including like receiving signals or sending signals, that kind of thing, of cell communication. But for our purposes today, the cell membrane it's kind of like our barrier between the outside and the inside of each individual cell. And without the functioning of the cell membrane, the organelles aren't going to be contained. And, you know, if you let too much stuff in, it could cause harm to the cell, or if you don't let enough stuff in, it could cause harm, so on and so forth. So it really controls what comes in and what goes out. And there's lots of reasons for that go down to a molecular level here. So most of the cell membrane consists of what we call the phospholipid bilayer. So take apart that first word there. Phospho is referring to phosphorus, and we learned about what lipids were in our first video. So lipids are primarily what make up the cell membrane. And here's the thing about the phospholipid bilayer. Through most of the cell membrane, unless there's special proteins that are going to allow other compounds to pass through into the cell or out of the cell, the phospholipid bilayer only allows small nonpolar substances to pass through. Okay, so this means that big molecules like glucose, proteins, they can't get through the phospholipid bilayer on their own. But small nonpolar molecules like, say, carbon dioxide or maybe even oxygen gas, they can pass through the phospholipid bilayer. Water, well, it can't, but it does. It doesn't go through the bilayer, but it goes through special channels that we'll talk about in our next video. But the phospholipid bilayer is going to be our main component of what we call the cell membrane. And we call it the phospholipid bilayer because it's got a phosphorus head, or a phosphate head, and then it's got what we call lipid tails. Okay, so these little squiggly lines, those are going to be lipids. And those are what's going to really be keeping out the polar substances, or the larger substances, from entering or leaving a cell. All right, so let's pretend that we're on a cell membrane right now. This is the outside of a cell. This is what's called the interstitial fluid. And then this is the inside of the cell, or the cytoplasm or cytosol of a cell. Okay, and we're going to have three different molecules in our little scenario over here. And the little hexagon is glucose, blue dots are water, and red dots are carbon dioxide. So if we've got the outside of the cell loaded up with each of these three compounds, okay, now there's a high, what we call, concentration on the outside of the phospholipid bilayer, the outside of the cell. Now, we just talked about a little bit what kind of molecules can pass through the phospholipid bilayer. So let's see what does. Oh, okay. Well, before we get to that, as I was saying before, it's highly concentrated on the outside and not concentrated on the inside, and that's what we call a concentration gradient. And a concentration gradient is established when there's more molecules on one side, or a higher concentration of them on one side than the other. All right, so in this scenario right here, we've got a lot on the outside and like none on the inside, even though it's not a real situation, but you get the idea. That's what we call a concentration gradient. Okay, so what molecules are going to be likely to do is they're going to kind of spread out and even out. Okay, but here's the thing. Water and glucose can't get through the barrier. Remember that? Glucose is too big, and water happens to be what we call a polar molecule. So it doesn't meet the cut, and it can't go through the bilayer. So carbon dioxide is the only one that's going to be spreading out nice and evenly between the two sides of the cell membrane. Okay, so carbon dioxide is going to diffuse to the other side of the membrane. So what happens is, well, yeah, because it's small and because it's non-polar. So voila, there it is. What's going to happen here? If we had a lot of carbon dioxide on the outside in a before scenario, it's going to spread out evenly. It's going to move what we call down the concentration gradient. I'll go over here. It's going to move down the concentration gradient so it kind of evenly spreads out over a little bit of time. That process, now I got to move my face, is called passive transport, or it's also called diffusion, where molecules naturally move from a higher concentration to a lower concentration without the use of energy. Okay, so imagine this scenario here. If I drop some food coloring into a glass of water, what's going to happen to the food coloring? Is it just going to stay as a little droplet the whole time while it's in the water? No, eventually it will spread out. And that's exactly what happens. That's called diffusion. That food coloring diffuses throughout that glass of water, and that's what's happening here. Okay? These molecules that are concentrated, imagine a little drop of food coloring, are going to spread out, and they're eventually going to be kind of even. They're going to be evenly diffused on either side of that membrane. It's just naturally going to happen. Molecules move from high concentration to low concentration. You're going to hear me say that until I'm blue in the face this year. High concentration to low concentration, all right? This is going to be really, really important when we talk about the urinary system, how your kidneys are able to filter out certain substances that should not be in your blood, all right? Or, maintain water balance. All right, so that's called diffusion or passive transport, when things are moving across the membrane without energy, and they just kind of move from high concentration to low concentration. Now, the opposite of passive transport is what we call active transport. So remember how carbon dioxide could move through the bilayer without any problems? Okay, not all molecules can do that, but your cells still need water, they still need glucose, and they can't get through that bilayer. So what's the way that it's able to do that? It can either move through like a channel protein, or the cell can use active transport. So large polar molecules get across the membrane through active transport, which requires energy. Okay, so hey, remember how I was going to say I was going to bring up ATP a bunch of times? Guess what? This is the first time I'm bringing up ATP after the lesson about ATP. So with a little extra energy that the cell has, it can use an ATP molecule, and it can force certain molecules, what's called against the concentration gradient. So with active transport, the cell can move substances from a low concentration to a high concentration. Okay, so a metaphor that's often used to compare passive transport and active transport, or diffusion and active transport, is that if it's passive transport, imagine you're canoeing downriver, downstream. Okay, you don't need a whole lot of energy to keep moving, right? But if you wanted to go upstream, or maybe like uphill on your bike or something like that, you're going to need some extra energy. You're going to need some extra energy to move in the opposite direction against the gradient. All right, so that's what active transport is all about. We're moving against the gradient, or we're moving molecules that can't get through the phospholipid bilayer. So check this out. This is kind of the same scenario as before and after, when we're discussing, let's see, I'll go right here, when we're discussing transport. All right, so if I've got an even distribution of these molecules on either side of the membrane, okay, my cell can bring up a little ATP and basically force these through a special channel. We can force these carbon dioxide molecules, or whatever molecules they are, out against the concentration gradient, right? So they're supposed to be naturally spreading out, right? But the cell can push them using a little ATP so that they have a higher concentration on the outside. So before we were going from high concentration to low concentration. This time we're going from low concentration to high concentration, and we need a little extra energy to do that. Okay, so as I said, active transport moves molecules against concentration gradients. Okay, so if we were to make a T-chart here, passive transport does not require energy, and it goes from high concentration to low concentration. Active transport does require energy, and it goes from low concentration to high concentration. So it goes in the opposite direction. Okay, so two other forms of active transport that we're going to talk about before we end up this lesson. It's called endocytosis and exocytosis. So the cell membrane, we're going to be doing a little activity on this later on, but the cell membrane is very, very fluid. It's almost like a bubble. In fact, a soap bubble that you can play with in the bath or whatever, um, it's really similar. That the structure of a bubble is very, very similar to that of a cell membrane. So imagine a bubble when you think of the cell membrane. And what the cell membrane is able to do, or what a cell is able to do with its cell membrane, is form smaller bubbles out of that bubble, just like bubbles, right? And the way it can do that, it can move molecules in smaller bubbles, or I should say, it can move bigger molecules inside of bubbles in and out by forming smaller bubbles. And the way it does this is through endocytosis and exocytosis. Endo, that prefix means in, and exo means out. So what is that? Endocytosis is the taking in of large particles by forming a vesicle. And a vesicle is like a smaller bubble. Okay, and here I have endocytosis, this image right over here. Okay, so let's just say this is like a protein or some kind of big molecule or something. Definitely can't pass through the bilayer by itself, right? It's too big. Okay, the bilayer doesn't allow really big molecules to pass through. So what does the cell do if it decides it needs that protein or whatever that big molecule is? It can form a bubble around it. It can change the shape of its cell membrane so that when it passes through, it has a little bubble on the inside of the cell, or that bubble is called a vesicle. All right, so that's endocytosis, but if it wants to do the opposite, and this is another type of, these are both types of active transport, by the way, they need energy in order to do this. Exocytosis can, if I want to take out some of these particles or a large particle, okay, my cell membrane can form a little membrane bubble, okay, and then pass it out to the outer cell membrane and then release it that way. So it doesn't have to pass through the bilayer, okay, that vesicle, that bubble becomes part of the cell membrane. Okay, and those are both types of active transport. All right, so that's what I'm going to be asking you to know for this video, the difference between passive transport and active transport, a little bit about the cell membrane, what it's going to let through and what it's not going to let through, and I'd like you to know a little bit about endocytosis and exocytosis as well. All right, so that'll be it for this video. Please get a hold of me if you have any questions, and we'll see you next time.